Well, hello, folks. Hope you're doing well today. We're going to be making that big drawing that incorporates all the different ideas from chapter 9 into one big visual document. Before we do that, I want to just review sort of the parts that are involved and then the processes that are involved, and then we'll put them together. So, again, I'm just going to basically run through a parts list and a process list, and then we'll have another video that has the big drawing. Okay, so in terms of the parts of the players that are involved, here are the energy molecules. Glucose, it's a 6 carbon molecule. And again, think of it as an energy molecule. There's energy in the bonds of carbohydrates. Pyruvate is a 3 carbon energy molecule created through glycolysis. When you split apart glucose, then you create 3 carbon molecules of pyruvate. NADH and FADH2, think of those as electron carriers. They strip away electrons and associated protons from food molecules, and then they transport them to the electron transport chain. And then ultimately, ATP is what the cell's going to make and what it's going to run on. Okay, other parts that are involved that aren't necessarily directly energy molecules, carbon dioxide, that is what gets split off when you break down carbs. Carbohydrates are carbon-based, so when you break them down, you split off that CO2, and then you exhale that. The cytosol, that's the fluid portion of the cell. That's where glycolysis happens, not in any specialized organelles, but just in the cytosol. The mitochondrion, that is the little squiggly organelle where the Krebs cycle and oxidative phosphorylation take place. That's where the majority of ATP in the body gets made. That's why it gets that nickname. Last couple of parts or players that are involved in this, ATP synthase. ACE tells it's an enzyme. Syn means together, to create. So ATP synthase is the enzyme that spins and then brings together ADP and then an inorganic phosphate, joins them together, and you get ATP. The cristae is the inner membrane of the mitochondrion, and that's where the ATP synthase is located. So that membrane is sort of a key spot for protein production. Now, in terms of the processes that are involved, first is glycolysis. Gly means sugar, lys means to split. So in the cytosol, we have this primitive ancient process where you take 6 carbon glucose and split it into 2 molecules of 3 carbon pyruvate. In the process, you get a couple of ATP out of it, and then you also make a couple of NADH molecules. You strip away protons and electrons from sugar, and then you put them in this carrier. Next is the Krebs or citric acid cycle. This happens inside the matrix, inside the fluid, inside the mitochondrion. So you have that link reaction where you take 3 carbon pyruvate and then you split it into a 2 carbon molecule. That 2 carbon molecule gets fed into the Krebs cycle. And in the process, the CO2 will get split off. You take that carbon, you split it off. And in doing so, you get, again, a couple of ATP, but you also strip away more protons and electrons and make more NADH and FADH2. So at this point, once you've completed the Krebs cycle, you've completely broken down your food and the carbon dioxide has been released, but you've only gotten like a net of 4 ATP out of it. Most of the energy, or a lot of the energy, is in NADH and FADH2. So now what do you do with that stuff? Well, you send NADH and FADH2, those electron carriers, to the electron transport chain. So NADH and FADH2 will drop off their passengers. They'll drop off the electrons that get passed down the electron transport chain. And then the protons get pumped into the intramembrane space and create a gradient. And a gradient like that is potential energy. Now, the last step here is oxidative phosphorylation. You use the potential energy in that gradient, and when the protons are flowing down their electrochemical gradient or their chemiosmotic gradient, it makes ATP synthase spin, and that powers the joining of ADP to phosphate to make ATP. All right, so that's an overview of the process. In the next clip, we're going to make the drawing. All right, thank you much.